Hey everyone, so today we're going to be talking about the cell in its entirety, right? So all those lovely little organelles in the cell and how it works. So in our last video, we went over the different types of cells and kind of the hierarchy, right? So cells turn into muscles, those muscles, sorry, did that a little backwards. So cells turn into tissues, those tissues will become mere muscles, muscles, the whole organism, right? So we also discuss, right, how cells are specialized and kind of tie into the characteristics of life, right? So these cells, how they're organized, right, they have different organelles. So today we're really going to be going into depth how those organelles work and kind of work together for that common purpose and the common good of the cell, and then in turn the whole of the multicellular organism. Alrighty, so before we get started, we want to go into the cell theory, right? So there are three parts to the cell theory. All life is made up of cells. Cells are the basic building blocks of life, and all cells come from pre-existing cells. Alrighty, so there are three primary scientists responsible for this. So you have Schleiden, who was a botanist at the time, right? So he was studying plants, and he had noticed that all plants had contained millions of little cells, right? So every single plant he looked at had these cells. He also happened to be good friends with Theodor Schwann, right? Who was not studying plants, but instead studying animals. So Schleiden had sent his work over to his buddy Schwann. Schwann had confirmed that he had seen a similar thing, but in animals. So this was all very interesting, right? And kind of helped lead Virchow into his overall cell theory. So he had kind of tied it all together and seen that all cells come from pre-existing cells, which was a really important discovery at this time. This is the 1800s, so really radical idea. Virchow was obviously very intelligent. He had some things really, really right? He had this cell theory that obviously had him with the idea that all life is made up of cells. They're the basic building block of life, and they have to come from one another. But he took this in a medical light, right? And just at that time, people were thinking that the fluids in people's bodies, they were calling humors. So you had a red humor, which was your blood, a yellow humor, which was your stomach bile, and then cancer, they were calling a black humor. So you may have heard of bloodletting before. So back in the day, right, if someone was very sick, they had thought something was malfunctioning with your humors. So what they would do was just make an incision and let that illness bleed out of you. Right? So seems very silly now, but back then, that was a great medical advancement that they thought was working. For whatever reason, I'm sure many, many people died. But Virchow, right, kind of was putting a stop to that, like, no, do not do that. There are cells responsible for this. It's got to be, right? If you are made up of cells, something must be malfunctioning with those cells. There must be germ cells out there, maybe that are, infecting your body, that are making you sick, that we want to try to get rid of. So he was very, very smart and really right about things. But on the flip side, he was really wrong about other things. He was not a fan of Charles Darwin. He thought Charles Darwin's work was nonsensical, didn't make any, no rhyme or reason to it. He did not believe in evolution or natural selection. So in some cases, right, you're not always right about everything. So sometimes it's good to listen to others. And the work of all these scientists, right, they are constantly building on one another. So that's one of the great things about science. The history of how we've gotten to where we've gotten today is really interesting. So I would definitely take a look. There's a funny YouTube video on the cell theory too. It's called The Wacky History of the Cell Theory. So learn a little bit more about those scientists if you want. Alright, so let's get into our cells now. right? So you have your prokaryote up here, very simple, no nucleus, right? They have free ribosomes, some circular DNA, all bundled up wherever, and then you have your eukaryotes. Right? So there's two different types of eukaryotes. You have your animal cell over here, and your plant cell. This should be reviewed from freshman year at this point, right? You want to be able to, one, know the difference between a eukaryote and prokaryote, but you also want to be able to differentiate between a plant and animal cell, right? Autotroph versus heterotroph. So just some key things there, right? A plant cell has a cell wall, right? Your animal cell will not. Plant cells have chloroplasts, animal cells do not. Both do have mitochondria. That's a common thing for people to forget, that your plant cell has that mitochondria, right? They need to utilize energy as well. Plants also have this large central vacuole. Sometimes you may see some people say it as a large central vesicle, and basically it just contains water to hold that pressure, right? Keep the cell walls nice and firm. Animal cells obviously do not need to utilize that, so they don't have that nice specialized structure. Animal cells, though, will have centrioles for cell division, so these aren't always here, they're only here during cell division. Plant cells don't have a use for that, they don't have them. Alrighty, so you want to be able to, rattle off some of the differences between plant and animal cell. You should be able to kind of recognize each of these structures and what they do pretty easily. So we will be doing a lab soon where you'll need to be able to identify and kind of recognize the difference between plant and animal cells. Alrighty, so let's get into the importance of organelles here. Right, so they are specialized structures. Right, so you have in your eukaryote all these specialized structures. Each organelle performs its own special function. Right, so depending on what type of cell you are and what your needs are, you're going to have different amounts of each organelle. Right, so a liver cell is not going to look exactly like a skin cell or a blood cell. Right, they're all going to have different amounts of organelles because they do different jobs. And they have different end goals. Righty, so these specialized structures are going to be really important, and the fact that each organelle can handle a certain job is going to be really beneficial for the cell, right? And it contains different things, right? So each organelle is going to contain different aspects, different chemicals, in order to make certain chemical reactions happen to carry out those processes. So think about, right, in your house, right? So your house is the cell. Right, you have each of these rooms, right, or containers, kind of like your organelles. So you have a kitchen, a bathroom in your house, a bedroom, right? Each of those rooms is serving a different purpose, right? So each is a specialized structure, right? So you're going to do your cooking in your kitchen. You're going to do bathroom stuff in your bathroom. You're going to sleep in your bedroom. Right, so if all of those things were combined into the same room, it would be chaotic, right? It would not be organized. You probably wouldn't be able to get much cooking done if, I don't know, Billy's on the toilet, right? It would not be a good scenario, right? So it is essential to keep them separate, right, to become most beneficial. Alrighty, and the cell feels the same way, right? So it has all these little organized structures in order to be most efficient. Alrighty, so what jobs do cells have? First and foremost, they make proteins, right? Proteins do everything for us. They create the structures. They're going to do all of the work. Alright, so in order for them to carry out these processes and do all the work, well, the cell needs to make some energy. Once you have those things, the cell can then make more cells, right? In order to survive and keep evolving, right? Keep your species going, you need to make more of yourself. So once all your needs are met, you're making proteins, you have energy, you're carrying out life on an efficient scale, you're going to make more of yourself. So whether you are whether you do sexual reproduction or asexual doesn't really make a difference. So here we have asexual reproduction, right? You start off with one cell, it's going to end up splitting into your two cells. Right, so in either case, you still need to make proteins, you still need to make energy in order to carry out your basic life purposes and your daily life functions. Right, so again, proteins do all of the work. They are so important. Right, so we have our DNA, which contains all of the information. Again, this should be hopefully a review from freshman year. So DNA has all your information. With that DNA, you can make your proteins. Right, so DNA is trapped in the nucleus. It has all the information. We don't want to be splitting up those blueprints, but we want to kind of make copies of ourselves and pass them out amongst the cell to make these proteins. The proteins will carry out all of the work for a cell, right? And once that cell is running efficiently, then you can make more cells. Once you have more cells, right, in some cases, we go on to make our multicellular organism, and it's just a big cycle. Alrighty, so some cell functions, right? How do we build these proteins that are essential for the cell doing its day-to-day thing? Well, we take that DNA, right? We want to make proteins, but that's not the only story, right? So if you probably remember from freshman year, right, we did transcription, translation, protein synthesis, all that lovely stuff, right? But then we kind of end there, right? So you have this protein, but how does it get to where it needs to go? Well, there's a couple more steps involved, right? And a couple more organelles which help make that happen. So once we make our protein, we have this primary structure, right? So this chain of amino acids is your primary structure. It then needs to get processed and folded. So we'll talk about where that happens in a minute. Eventually it becomes your tertiary structure. So again, it's just folded more thoroughly into this tertiary structure or quaternary structure. Once it is all folded up, packaged into a nice little box, then it can be delivered and shipped to where it needs to be used either inside of the cell or it can be shipped outside of the cell to be utilized as well. Alright, so how do we build these proteins? Alright, so there are some organelles involved. You have your nucleus, right, which contains the DNA. We're going to make a copy of that blueprint called mRNA and send it on out to some ribosomes. The ribosomes can either be found free in the cytoplasm, or if we're shipping proteins outside of the cell, those ribosomes will be on the endoplasmic reticulum. Alrighty, so it's called the rough endoplasmic reticulum when the ribosomes are on there. Once we're done with the endoplasmic reticulum, right, they will head to the Golgi apparatus. The Golgi apparatus will make a final vesicle to send it out of the cell. Alrighty, so you want to kind of keep in mind this chain or assembly line as we're going through what each of these organelles does a little bit more in depth. Alrighty, so first we have our nucleus, right? So really important guy. He's going to protect and store all of your DNA, right? So here you see our DNA. There's also this large, well, it looks purple in this illustration, and we'll talk about what that is in a minute. It's called the nucleolus, right? But right now we just want to focus on the overall structure of the nucleus, right? So there's this nuclear envelope. So right around here you see it. It's going to be, a double membrane, right? But also there's these small little pores or holes that we want to make sure are small enough so that way the DNA, right, that very important and vital DNA doesn't get out of the nucleus and it's protected, but we do need it large enough so that way the copies or our mRNA can get out of it, right? So we do want the mRNA to be able to escape the nucleus, right, get out of those pores so it can make protein. And we also need, so there are ribosomes which are made inside of that nucleus that need to escape through those pores as well and get out into the cell. So that pore size is going to be extremely important because it can't be too big where DNA can escape, but it needs to be big enough where other things can get out. Alrighty, so this should jog some memories, right? Lovely protein synthesis. You have your DNA. It's being copied during transcription to make mRNA. From there, it gets edited. Only exons will be put together to kind of exit through a nuclear pore. From there, translation occurs. It'll meet up with the mRNA is translated with your tRNA and with a ribosome. Right, so that ribosome can either be free in the cytoplasm or it'll be on the endoplasmic reticulum to start making your protein. Alrighty, so where did these ribosomes come from? Well, they were made in the nucleolus. So the nucleolus actually isn't a physical thing. It often just looks darker in images because it's a very active site. So there's constantly ribosomes being made there. So it appears darker when we take an image of it. So those ribosomes that are made exit through the nuclear pores and combine to form functional ribosomes. So what I mean by that, right, so a lot of times people think of just the ribosome as one big unit. It's actually two units that are together. So it's a large subunit and a small subunit. Alrighty, so here are our ribosomes. So they help in protein production. It's that large subunit and small unit which have come together with the mRNA. So remember the tRNA will come in and drop off those amino acids to build your protein. Again, they could be found free in the cytoplasm or on the endoplasmic reticulum. So these little dots here are those ribosomes on the rough ER. Alright, so our endoplasmic reticulum, right, is going to process proteins, manufacture membranes, and also synthesize and hydrolyze many compounds. So remember, there's two types of endoplasmic reticulum, right? So there's that rough endoplasmic reticulum, which is going to be responsible for processing proteins, but there's also smooth endoplasmic reticulum, which is going to be primarily responsible for these two, right? Manufacturing membranes and synthesizing and hydrolyzing compounds. So the structure of an endoplasmic reticulum, typically it'll be directly attached to that nuclear envelope and it'll extend throughout the cell. Alrighty, so let's take a look. Right, so here's our nucleus, right? And then you have your rough endoplasmic reticulum. I know it's the rough endoplasmic because you see these little dots here. Those are your ribosomes. Right? And then you have your smooth endoplasmic reticulum a little bit later on. Right, so depending on what type of cell you are and what the needs are of that cell will depend on how much of each of these you have. Okay, and you could see, right, it's directly connected to that nuclear envelope. Right, we want this process to be efficient. We need proteins to do all of the work. So as soon as that information from the DNA gets sent out, we want to translate it right away. We want to start making that protein. Alrighty, so our smooth endoplasmic reticulum, right, again, we said membrane production. It'll do many of our metabolic processes. How it does that, oops, sorry, skipping ahead, is it's going to synthesize, right, or put together different lipids, right, so these lipids could be like oils, phospholipids. Remember, phospholipids are, we have a phospholipid bilayer, different steroids and sex hormones. Smooth endoplasmic reticulum are responsible for creating. The smooth ER also hydrolyzes many chemical structures, right? So it's going to hydrolyze glycogen into glucose. So this happens in the liver. It also detoxifies drugs and poisons in the liver. So examples would be like alcohol and barbiturates. Right, so your liver cell is going to contain a lot of smooth endoplasmic reticulum, whereas maybe your skin cell really wouldn't need to hydrolyze all that much. So maybe it wouldn't have as much smooth endoplasmic reticulum. Alrighty, whereas the rough endoplasmic reticulum, it's going to produce our proteins. Like we said, right, it has the ribosomes, so that once it's done making that protein, it travels through the endoplasmic reticulum and then can send it off to the Golgi apparatus to be exported out of the cell. Alrighty, so you have your protein secreting cells. It also packages into transport vesicles for export. So we see here it transporting it into vesicles, kind of go into the next phase or the next organelle. Cells that would have, so just to bring up, like we did with the smoothie R for cell specialization, a cell like your pancreas cell, right? So cells in your pancreas would need a ton of rough ER because they are making a lot of enzymes, right? So they're constantly making enzymes to help with digestion. So enzymes, hopefully you remember from freshman year, are proteins, right? So since we need to create so much of these proteins, you're going to have a lot of rough endoplasmic reticulum. Alrighty, so here's just a close-up shot of the endoplasmic reticulum, right? You have your ribosome attached on the outside, that protein which was made. That protein, once it's done being folded up out of its primary structure, it's going to head into a vesicle. That vesicle now is going to head to the Golgi apparatus. Alrighty, so you have your Golgi apparatus. Its function for the Golgi is going to finish, sort, tag, and ship. It's kind of like a mail room, UPS room, I don't know, Amazon shipping department, whatever you want to think of it as. So it's going to ship products in these vesicles, right? So it kind of looks like a membrane of Sachs. Right, so from that endoplasmic reticulum, it came in on the cis phase. Right, so it becomes the cis Golgi vesicle. From there, it enters the Golgi, where it is going to be sorted, tagged, kind of finished folding up and putting a nice bow on it. And then once we're done with the Golgi apparatus, a final vesicle is formed on the trans phase. You have your trans vesicle now, where it's going to be transported out of the cell. All right, so you have, again, your Golgi apparatus, right, so your endoplasmic reticulum. Again, this process is for if we're making a protein that needs to be shipped out. All right, so if it's a protein that's going to stay in the cell, it would probably be on a free ribosome. That would be, we would make the protein out there in the cytoplasm. So here, if we want to ship out a protein, we would go through the endoplasmic reticulum. Once done with the endoplasmic reticulum, you would form a vesicle on the cis Golgi, head into the Golgi apparatus, and then that Golgi would make a transport vesicle or trans Golgi to head out into the cell. Right, so I always remember it as, here's another picture. Right, so you have your endoplasmic reticulum, rough, ER, your ribosomes, protein was made. This is the cis vesicle. There it goes. Protein's nice and folded up, and then you have your trans vesicle transporting it out. Right, so I always tend to remember the cis side versus trans side if you think about the alphabet. Right, so cis starts with a C, comes earlier in the alphabet, so it's going to come over here. Later on in the alphabet, you have a T for your trans side. So you can also think of it, obviously, transport, right? So it's going to transport those proteins outside of the cell. So that's kind of just wrapping it all up. Alrighty, and finally, we're going to talk about the evolution. How did all these organelles get here? So you have Lynn Margulis, a very important scientist who came up with the endosymbiotic theory. Right, so she believed we started off as this ancestral prokaryote. Right, so you had a plasma membrane, DNA kind of floating aimlessly, no nucleus or anything, and some free ribosomes. Then, as time continued, right, this ancestral prokaryote started infolding or causing invagination. Sometimes you'll see it as. So it started closing in on itself, and then finally it pinched off to create these little pockets, right? Or eventually what would become some of our organelles, right? The endoplasmic reticulum, maybe Golgi apparatus, right? All of these kind of little infoldings. So now you have a pretty big bacteria with a nucleus, maybe doing some basic functions. So then what happened next, they're not sure if this happened on purpose, right? Was this one very large cell trying to consume a mitochondria, since these mitochondria, right, they believe were, or she believed, was its own separate prokaryote, right? So they have their own DNA, they were living freely. So they're not sure if this larger cell was trying to just consume a mitochondria, but the endosymbiotic theory believes that this cell engulfed the mitochondria, and then they ended up kind of coexisting, right? They were helping each other out. So symbiosis, both were benefiting. For whatever reason, the cell didn't destroy it and use it. They coexisted. The cell now was able to utilize the energy that the mitochondria was creating for it. Then, right, we kind of take a little detour here, and some of the cells end up doing a similar thing to chloroplast. So chloroplast was also its own free-living organism at one point, right? And it also got engulfed, and for whatever reason was not destroyed. They end up coexisting, right? And then these will create your autotrophs, right? So here we have our heterotrophs and our autotrophs. So there are two main reasons why people agree with this theory, right? So one of the reasons being, which I had mentioned before, was that mitochondria and chloroplasts alike have their own DNA. So they have their own circular DNA, which is very similar to our current day bacterial DNA, right? So they may have at one point been living on their own, right? So the second point is that they have a double membrane. So none of our other organelles have this double membrane going on, but for whatever reason, the mitochondria does, right? So it has its own, one, double membrane, and two, its own special DNA, right? So when our cells go to divide, these mitochondria actually will divide on their own and do their own thing, versus your other organelles, which kind of just like get bigger and stretch and then split with the cell, right? So they're doing their own thing primarily. So that's kind of all the reasons why people agree with Lynn Margulis and her endosymbiotic theory. Alrighty, so that's all I have for you today.